Hello everyone, my name is Xiaoxiao Xiao, a PhD candidate in medical technology at the School of Medical Imaging at Tianjin Medical University, China. It's my pleasure to present my research on assessing the effect of ComBat for correcting scanner effects of multi-modal brain MRI metrics. There will be three main points in my presentation: the background and objectives of my study, the analysis I conducted, and finally the summary of this research. Now let's begin with the background and objectives. Over the past 3 decades, advancements in neuroimaging have led to the accumulation of numerous multi-center large datasets. Large data sample can enhance statistical power and help us obtain more reliable results. But using multi-center data directly has its drawbacks. Different centers use different MR scanners with the differences in manufacturers, feed strength, etc., leading to heterogeneity in brain imaging data. This heterogeneity may obscure the real intergroup differences or even amplify the intergroup differences. Previous studies have reported the interscanner heterogeneity of multimodal brain metrics, and some have attempted to correct it with various methods. But these studies have some limitations. Firstly, some studies have no repeatedly connected data, therefore the assessment of interscanner heterogeneity may include individual variability. And additionally, the number of MR scanners involved in these studies is usually limited, and most of the studies are based on single modality data, and they cannot comprehensively characterize the interscanner heterogeneity of multiple modalities. To address these issues, we conducted two analyses based on the data of imaging consortia. The first analysis utilized multi-modal brain MRI data repeatedly acquired on 28 MR scanners to characterize the interscanner heterogeneity of multi-modal brain metrics, both before and after data harmonization. We then investigated the impact of heterogeneity on brain behavioral association, as well as the effect of data harmonization on improving the detection of brain behavioral association by using the multi-modal brain MRI and behavioral data for approximately 7,000 subjects. Now let's focus on the analysis. The first analysis. The first analysis involved 28 MR scanners from 3 different manufacturers, from which the data of 5 commonly used modalities were collected, and then brain region and voxel level metrics were extracted for subsequent analysis. We chose ComBat to correct interscanner heterogeneity as it performs well with small sample data, and it has many advantages over other methods. This benefit has led to the widespread use of ComBat across multiple modalities. We then used the intra-class correlation coefficient to quantify the interscanner heterogeneity of multimodal brain MR metrics, lower ICC indicating higher level of heterogeneity. Here are the results. Due to the large number of metrics analyzed in our study, only a subset of them are presented. The ICCs of metrics are represented as colored bars on a polar coordinate plot. Each bar represents a single metric, with the outer labels correspond to the names of the metrics. The darker bars indicate the ICCs prior to ComBat harmonization, and the lighter bars correspond to the ICCs after ComBat harmonization. These are metrics for resting state fMRI, DTI, and DKI, and these metrics were derived from free surface segmentation. These are metrics for brain network and functional connectivity. These are metrics for functional connectivity density and strength. To summarize the results of the first analysis, We get to know that all metrics exhibit interscanner heterogeneity, and ComBat can correct interscanner heterogeneity for most of the metrics. Now that all metrics exhibit different degrees of interscanner heterogeneity, it's important to understand the impact of heterogeneity on scientific studies. To clarify this, we conducted a second analysis. To investigate the impact of interscanner heterogeneity on brain behavior association, as well as the effect of ComBat harmonization for improving the detection of brain behavior association by using the data of approximately 7,000 subjects. Taking voxel level metrics for an example, we selected 10 metrics with their ICCs ranging from about 0 to 0.9 in increments of 0.1. We then computed the correlations between these 10 metrics and 191 behavioral variables. Typically, there are two approaches of conducting association analysis. The first one is mega-analysis, which involves combining data from multiple scanners and then conducting association analysis with or without ComBat harmonization. The other one is meta-analysis, which involves conducting association analysis with the data from each scanner and then combining the effect sizes. We evaluate the impact of heterogeneity on mega-analysis by comparing the result of mega-analysis with or without ComBat harmonization with the result of meta-analysis as a reference, which is thought to be reliable. We then evaluated the impact of interscanner heterogeneity from 5 different aspects, but due to time constraints, we will only discuss 2 of them. On the 2 figures presented, the percentages of significant associations and the overlap between meta and mega-analysis. We can see that the blue bars represent the results of mega-analysis without ComBat harmonization, denoted as pre-ComBat, and the cyan bars represent the result of mega-analysis with ComBat harmonization, denoted as post-ComBat, and the orange bars represent the results of meta-analysis. These results are based on the behavior variable that associated with the maximum number of brain voxels. According to the results, pre- and post-ComBat is almost identical, indicating that the impact of heterogeneity is minimal. But when the behavior variable that associated with less than 10% of the brain voxels was selected for conducting the association analysis, the results differed. It can be observed that when the ICCs were greater than or equal to 0.4, pre-ComBat had more significant associations but had lower overlap with meta-analysis. To summarize the results of the second analysis, we know that when the behavior variable associated with most of the brain voxels, whatever the ICCs were, the interscanner heterogeneity can only slightly impact the brain behavior association. But when the behavior variable associated with the limited number of brain voxels, 10% in our study, interscanner heterogeneity could result in an excess amount of unreliable significant associations, which could be reduced by ComBat harmonization. This is when the ICCs were greater than or equal to 0.4, but when the ICCs were less than 0.4, ComBat didn't perform well to correct interscanner heterogeneity. To conclude, the following is the summary of the advantages and limitations of the study. First, there were 28 MR scanners in our study, and we used repeatedly collecting multi-modal data in Part 1 and a larger sample size in Part 2. But there were only 2 subjects in Part 1, limiting the ability to perform group-level analysis, and we only analyzed 10 metrics in Part 2 due to their time-consuming and storage-consuming nature. That concludes my presentation. Thanks for your attention. And these are the disclosures.